So for today's lab, we're going to be talking about exercise 12, which is antibiotic response. So why would we do this? Why would we test antibiotics? Well, the thing that we're trying to determine is which antibiotic is successful at eliminating certain bacteria. We're trying to test the bacteria's ability to resist the antibiotic. It's a way to basically make sure that the antibiotics chosen are actually effective against what the bacteria is that's, let's say, causing an infection. So things to consider. As mentioned in the lecture, we talked about the fact that antibiotics are going to be things that are going to attack specifically bacteria. They're not effective against viruses. If you go into the doctor because you think you have the flu, they should never give you antibiotics because the flu is a virus. Antibiotics don't work on those. We do have different types of antibiotics that work in different ways, making them more effective against certain bacteria compared to others. We talked about narrow spectrum antibiotics versus broad spectrum antibiotics. So an example of a narrow spectrum antibiotic would be something like isoniazid. Isoniazid is one type of antibiotic, but it is only effective against mycobacterial species. So if you're trying to treat something that's gram-negative or gram-positive or something else, it may not work because it is very narrow spectrum. It only works on this one species. Broad spectrum antibiotics tend to have a wider reach as far as things that it would be able to treat. An example of a broad spectrum antibiotic would be something that would treat both gram-positive and gram-negative, as well as maybe intracellular pathogens or even pathogens that don't have a cell wall at all. If you have one medication that treats all of those varieties, then you have something with a very broad reach, broad spectrum, that can treat quite a few diseases all at once. When doctors are picking a regimen for treatment, these are things to consider. So for example, that broad spectrum antibiotic that I just made up, if I have a patient and I haven't figured out what they're sick from yet, I would put them on a broad spectrum antibiotic at least at the beginning, something that I know treats a wide variety of things. That gives them more of an opportunity of hopefully having something that's effective. If I keep taking samples from that patient and eventually figure out exactly what it is that's infecting them, then I can change the treatment to something that is specific for that bacteria. But until I know that, broad spectrum makes the most sense because it's the thing with the widest reach that can treat the most bacteria. So how do I choose the right treatment? As a microbiologist, there's a simple test that we can use called the Kirby-Bauer technique. You will need bacteria, you will need a nutrient agar plate, and you'll need antibiotic discs. Basically little paper discs that have been infused with the antibiotic. Each one will have a unique marker to identify which antibiotic that disc is infused with. And what you are going to do is you are going to observe how much clearance there is around that disc. So if this is a plate that has been carpeted with bacteria, which I'll show you how to do in just a minute, then you put your antibiotic discs in place. You can see that with this disc, FM300, there's barely any clearance around that disc, meaning that this specific antibiotic probably isn't all that great against whatever bacteria is on this plate. However, if you look at CRO30, look at how big that clearance is. That's a huge amount of clearance around that particular disc. So admitted, I don't know if this is enough clearance to actually say, oh yes, this is showing very good antibiotic control of this bacteria. because there's a way that you actually have to measure it, which I'll get into in just a moment. But you can definitely see a difference between this clearance versus this clearance. So what we're going to do is we are going to create a carpet streak plate today. We're going to take different antibiotic infused discs and we're going to embed them into that agar that we've just carpeted with bacteria. Then we'll let them incubate for 48 hours. After that 48 hours, so after 48 hours of incubation, you're going to again take your plate out and you're going to observe the bacterial reactions to each specific antibiotic. There are two possible outcomes here, and you can actually see two really good examples of this. G, where there's no clearance at all, and then you can see that pretty much everybody else has some type of area of clearance, but the sizes can vary. All right, so if we have a reaction like G here, what does it mean? Well, ultimately, what it's going to mean is that it is not an effective antibiotic for that specific bacteria. There's no area that is clear of bacteria, which means it didn't work because the bacteria grew all the way up to the edge of that disc. Now, if you look at B here, if there is an area around the disc with no bacterial growth, what does that mean? Well, in this case, well, actually all of these cases, it means that I have an area that that bacteria can't grow because the antibiotic is inhibiting it from working, from basically being able to live in that space. So what do we call this space where there's no bacteria growing? Well, if you think about what the antibiotic is doing, it's inhibiting the growth of that bacteria. So we call this space here the area of inhibition. It's inhibiting the bacteria from growing. Now, what we are going to do is we are going to take a ruler and dead center in the middle of this disc, we're going to measure from end to end in millimeters. How many millimeters is that? Once we have that number, that measurement, why would it be significant? Well, the bigger the area of inhibition, the more effective that antibiotic is. How do we tell if it's actually effective enough to treat an infection with this specific bacteria? Well, we actually have a chart called a Kirby-Bauer chart. Remember, this is the Kirby-Bauer test. So this Kirby-Bauer chart looks something like this. We've got the antibiotics listed, at least the ones that I have are highlighted in green. And then you've got the amount of milligrams or micrograms that are in that disc infused in it. And you have whether it is resistant, whether it is intermediate, it kind of works, but not that great, or if the actual bacteria is susceptible to that antibiotic. So I'm going to zoom in a little bit. So in this example, I have ampicillin. Ampicillin works against gram-negative organisms and enterococci or staphylococci and penicillin susceptibles. Now, here's the deal though. Remember I said that measurement is really important. One of the reasons that I chose this specific antibiotic is because whether or not I'm working on this type of organism or this type of organism, it actually varies how big that measurement has to be. Okay, gram-negative organisms and enterococci. If I have less than 12 millimeters, then it means it is resistant to ampicillin. If I have between 12 and 13 millimeters, it's kind of intermediate. It works okay, but it's not the best. However, if I have an area of inhibition that is actually bigger than 13 millimeters, that means that that specific bacteria is susceptible. Now remember, this is for gram-negative organisms and enterococci. If I'm looking at anything that's staphylococci or the penicillin susceptible ones, it has to be greater than, I'm going to go over here, greater than 28 millimeters for it to be considered susceptible. Intermediate is between 21 and 28, whereas resistant is anything less than 21. Now, if you look, that less than 21 is even bigger than what we see with the gram-negative organisms. So when we are looking at that area of inhibition, we have to have the chart, because the chart is going to show us whether that measurement is significant or isn't significant. And in some cases, what might be significant for one species of bacteria may not be for another. Again, this is why the chart is so important. The next video is going to go through how to actually do the experiment itself.